about drug delivery. And I don't think we need to incentivize people why this problem is important, because you've been through CRISPR revolution, AAV delivery from COVID vaccine. So we think it's a trending topic right now that is very important for everybody to know its basic principles, how it works, what's the challenges, and where we can move on. And today we are very happy to invite two speakers. The first speaker is Jason Yanjun Lu. So first he will give us a talk about his work, and then we'll have Dr. Yunzu Chen join us in a panel discussion after Jason's talk. And people online, feel free to raise your hand if you have questions, and totally okay to interrupt the talk if you have any questions. So without further ado, let me pass down to Jason.
Thank you for a nice introduction, and good afternoon, everyone. So I'm Jason, and today I'm going to be here to share some of my research and the idea concept of the drug delivery. So before I start, I just want to ask, so what's the background here for today's attendees? Are you the biologists? Can you just raise your hand? Engineer? Chemist? And the rest is computation. Okay, yeah. So it's kind of very interesting topic for the drug delivery. And today I'm going to just talk about the concept, we already have the pharmaceutical company and biotech company, and they are developing a new drug, but why do we need a drug delivery? But before I start, just first introduce myself. So I have pretty diverse background. I received my bachelor degree in chemical engineering and civil engineering from National Taiwan University. And I moved to state and went to Cornell University for my master, my PhD in biological engineering. So in first half, my the PhD research still apply the engineering principle in the biological, the biomanufacturing and also the biological applications. And after received my PhD, I moved to Boston to join the Langer Anderson Lab and Boston Children's Hospital and the MIT. So you can see I have a very diverse background, receive a training from engineering, chemistry, and also the biology. And it's really great time to join the Langer Anderson Lab at MIT, because this Bob and Dan really give me a lot of the support and resource. For example, after 3 months joining the MIT, I already have a thorough report Dr. Han to help me on a lot of my projects. Also, I know the chemistry, but I'm not the expert in the synthetic chemistry. So I need someone to help me after I design the materials, she'll be the one to help me to do all the synthesis. And since I have pretty diverse background, so I just define my research experience by the size scale, nanoscale, microscale, and the macro scale. So in the nanoscales, major on my major work at MIT, developing the nanomedicines. So first is people will be pretty familiar with is lipid nanoparticles. So after COVID pandemic, the mRNA therapeutic become pretty popular because that's the COVID vaccine. And why it's pretty popular because we got the FDA approval, get the EUA for the vaccine within just 1 year. So that's kind of we break the record in the mRNA technologies. And the second one is I work with one of my friends who's a faculty at Joslin Diabetes Centers to develop the mRNA therapeutics for the obesity and type 2 diabetes. And the third project I did is work with a chemist in the lab to develop a degradable material for mRNA deliveries. And on the bottom left, that's you develop some using the polymer-based the materials to do a peptide or protein delivery. For example, on the right, that's for the glucagon deliveries. We all know glucagon is pretty unstable in the body, so the half-time for the glucagon in the body probably just 3 minutes. So how we improve the PK and improve the efficacy in the patient. So I developed a new material to combine with the glucose and the POJ all together to encapsulate the glucagon, so we can protect the glucagon until it reaches to the liver, because we know like the liver is the major organ for the glucagons. And when we jump to the micro scale, that's my PhD research. So as I mentioned, in the first the research project when I joined the PhD program at Cornell, it's I developed a new the method to do the 3D the encapsulations. So the traditional 3D cell culture, people pretty familiar with it. It's we can just simply mixing the hydrogel with or extracellular matrix with the cell and dripping into the petri dish, well plate, or any the hand drop layer. But the problem is we need a lot of the labor intense work, because if we need to do high throughput screening, we need the 1,000 the 3D for example tumor spheroid. Then that means we probably need to hire 3 technicians just dripping the plate every day from A to 5. So that's kind of not the smart way to work on this project. So one time, what I did is to transfer this into the hydrogel systems. So inside the hydrogel capsules, we can put any type of extracellular matrix to support the cellular growth and cellular functions. And the green color is the alginate. That's a hydrogel. So the great thing for using the hydrogel, because the pore size of the hydrogel is small enough to prevent any the immune system attack to protect the cell from the shear force, but also large enough to allow a nutrient, oxygen, or even the therapeutic peptides to diffuse in and out from the capsules. So since it has the way to protect the cell from the shear force, so we can, if we want to do a biomanufacturing, we can just move these capsules into the stir bioreactors, because not every cell is sustained for the shear force. For example, the stem cells. And it can also protect the cell from immune systems. That means if we want to use cell as a therapeutics, we can definitely use it, because it can protect the cell from the immune attack. So for example, if we are putting the islet cell in the hydrogel capsules, we can implant into the Taiwan diabetes patients to treat, help them to even just cure the diabetes. So and how we make this the capsule system, I borrow a pretty old technology called the electrospraying. So it's an old technology people use for printers. And what I did is design a special nozzles, and the outer layer inject the hydrogel, that's a green colors. And the middle layer is we can put any type of cells and any type of extracellular matrix, and just put a high voltage supplier to create the electric field to drag or dripping all the solutions into the cross-linking baths to form the hydrogels and encapsulate the cells inside the capsules. So we can see we have pretty uniform the capsules, and the beauty of this technology is we can make more than 10,000 capsules in a minute. So that means if each capsule is a mini bioreactors we want to use for drug screening, that means we can make more than 10,000 capsules or 10,000 mini bioreactors in a minute. And inside of the capsules, here, because the red color is the cancer cell line, the MDA and B231, and we are putting the type 1 collagen fibers to support the cell growth and cell functions, because we all know cancer cell likes the type 1 collagen fibers and use it to migrate and to get the growth signals as well. So here we use the confocal microscope to confirm the structure. The inside the core is a pure collagen fibers, and it's interacting with the cell to support the cellular growth. And another interesting is using these methods, we can also control the flow rate to control the core size. So in order, we can control the size of the tumor aggregates or any type of the microtissues. And not just this structure, I also develop different structure for different purpose. But here just a demonstration how many structure we can do for using the electrospraying technique to encapsulate the cells. And applications, that's something we are talking about. If we are using the cell as a therapeutics. So that's another example. Is like if we encapsulate the mesenchymal stem cell, because we know like the mesenchymal stem cell will secreting a lot of microphysical and can help the wound healing, like the fibroblasts to migrate to close the wound. So that's what we did to encapsulate the mesenchymal stem cell and try to implant into the horse to see if we can just help the horse to improve or enhance the wound healings. And also we use for like the hepatocyte, like the encapsulations, because we know like the hepatocytes will be a problem. Like if we do collect the hepatocyte from patients or from any animals, and it's hard to immortalize the hepatocytes. And what we did is like we co-culture the hepatocyte with the stromal fibroblasts and encapsulate into the hydrogel capsules and implant into the mice again. So in the future, if the stem cell technology work well, there might be a new way for us for like the future, like the liver transplantations. And the last is like we use it for the cell segregation studies. And another interesting is like organoids pretty popular since like 2015, because like the organoid, what we call is like called like the mini organs. So by using this technique, we can do the incorporate into the automation like the high throughput like production system. So we can encapsulate the organoid and do the cell culture and put into the stir bioreactors to improve the mass transfer and the heat transfers. So we can use this method to highly to improve the expansion for the organoid as well. So that's a quick like study. We use mice like small intestine organoids, and we found like using our capsule system, we can improve the expansion rate by 1.5 times. And the last is like, I also did for some like the basic research. So using the same system to mimic like how the breast cancer formations, because that's some research shown like when some women have like higher mammographic density, these people has a higher chance to get like the breast cancers. And we are trying to study how the stiff the micro environment can like induce like the normal like memory epithelial cells to transform into the tumors. So what we did here is like we use the alginate to mimic the stiff or dense fiber environment, and inside the capsules we put the basement like the membrane matrix, the matrix to mimic the basement membranes here, to actually to mimic the memory duct formations. And what we use here is like use the MCF10A cells, because like the MCF10A cell is the benign like the human memory epithelial cells, and if we just mix them, culture them in the matrix, within like the 5 days, it can form a pretty nice polarized like hollow structures, and people use it to mimic like the memory tube. And we found like when we just encapsulate the cells into the hydrogel capsules, even just one single cells, this cell will just crazy growing, and they will fill the core pretty quickly within like 3 days, and then you'll keep growing and pushing the hydrogel, and eventually you'll find a weak spot to break the capsule and escape from the capsule. That's total. To me, it's like almost similar to like the cancer status, metastatus, and this makes us pretty interesting. And we are trying to see like if this is just simple like the physical ways, is it actually to malignant transform the cells? So we collect the cell from the capsules and inject into the memory fat in the skip edge mice to see like if they can form the tumors. And we also did like the RNA-seq to confirm like how the physical environment will affect the cells. And we found like there are more than like the 6,000 gene was significant change by just simply by the physical confinements here. And the last is like, I also work on some like the macro device. So that's also one thing that I will just focus on in the following slides. So I've mentioned about like we can put the islet cell in the capsule and implant into the Taiwan diabetes patients. advantage using a capsule is that it's easy to implant, but we cannot like expect a cell can have function and survive forever in the body. So at a certain point, we need to find a way to replace the cell and to keep our like a therapeutic drug. Here we're using the cells to keep like producing the insulin or glucagon for our Taiwan diabetes patients. So we developing like some like a macro scale like a device, so we can easily to retrieve the device and replace the cell and reimplant into the patients again. And in the middle, it's like I work with a postdoc who's like training as like an electrical engineer. So we are thinking about like a lot of like in the drug delivery, people use the chemical way to do the drug response, to use chemical response to do a drug release. But for some drug, for example, glucagon, why we need a glucagon? Because like the Taiwan diabetes patients has some like the low glucose like conditions, and in the hypoglycemia, like the patient might have DC or even die. So they need a glucagon injection immediately. So the current way people do is like they need to dissolve the glucagon in the solvent and inject into the body as soon as possible. And what we're doing here is like we're thinking about like we have pretty nice like the wireless like technology. Why can't we just use the wireless technology to help us to do the precise like drug delivery? So what we are thinking about is like if we can connect the CGM, the continuous glucose meters, with our like a drug release device. So if any like the CGM detect the low glucose like the conditions, you are wireless to order our device to break the capsules and also release the drug into the patient's bodies. And the last part is for fun. So at MIT, I also work with artists. So we are trying to recreate like an artificial ear for Vincent van Gogh. So people know like Vincent van Gogh has like a depression, so he cut his ear. So we can see like that's his self-portrait. Like he didn't have his like the right ear. So we are trying to do is like we're trying to recreate his ear. So we found like his brother's like the grandson and flew to Boston to donate his chondrocyte. So we immortalized it and use a 3D printer scaffold to scan like his descendants' ear, and we just print out using a biomaterial to print out like the ear scaffold and see those cells into the scaffolds. And those is like we culture them in the bioreactors. So this is our project. Like we have shown this in Morris Art Museum in 2018, 2019 in Japan. So currently, like I work at a biotech company called modality therapeutics. So we are like the antibody companies. So we have the MSTAR platform for the multi-specific antibody. So as we can see, we can up to like the 6 binding sites in our like the antibody designs. And that's what we use for a lot of different like applications. For example, like we use our multi-specific antibody for the coffee, like the prevention, because we know like coffee have mutations. If we have like our like one like antibody can cover all the mutation sites, we can just use one antibody for the prevention or for the treatment purpose. But I was hired to help them to establish the whole nucleic acid delivery platform, including the DNA, RNA. So I also need to bring in the lipid library for them for the LNP for the mRNA delivery or DNA delivery. Also, I developed a new technique, or also that's the current trend for the mRNA research. Like I combine with the antibody technology with the LNP, so we can use the antibody as a targeting molecule to help us to do the targeting delivery. For example, if we want to target to the lung, we can just find the right antibody to help us to lead out like the LNP to target the lung cells or lung endothelial cells. Okay, so that's pretty quick like introduction for myself because I have pretty diverse background. And if you have more questions, I'm happy to answer any question from not about like the drug delivery. So since today's topic is drug delivery, so what's the first thing? That's first thing is the drug. So we have like different like drug modalities. So small molecule, large molecule means like the protein, peptide, and current like antibody become like also pretty popular now. And since 2017, gene therapy has also become pretty popular. How we use the DNA or mRNA as like the therapeutics. And the last one is the cell therapy, for example. People probably pretty familiar with cell therapy will be the CAR T cell therapy for the like cancer treatments. But today I'm going to talk about different things like the cell replacement therapy, for example, what I just mentioned using the pancreatic cells for the like Taiwan diabetes treatments. And first, like we have a lot of different drugs. The second thing is like how we do the delivery. For example, like we can have like the proteins for the drug delivery, but why in the past like a lot of like the biotech or pharmaceutical company doing the clinical trial, they pretty easy to fail the clinical because like sometimes like we have the right molecule, but we cannot deliver to the right place or to the right cells. So that's why like we have the drug delivery system we want to do like to use for our like the targeting like deliveries. And that's a different like for the drug delivery, that's pretty broad like terms. So for example, you can say like, for example, for glucagon delivery, because glucagon is pretty unstable, so we need to find a way to protect the glucagons. That's a drug delivery. But if we want to do the local deliveries, so for example, microneedle patch will be also a method we can use for the drug delivery. And for the nucleic acid lipid nanoparticles, a new like ionizable lipids to like encapsulate the mRNA or DNA and deliver to the right like the cell or the organs. That's drug delivery. So it's pretty broad like the terms. And today I'm not going to go pretty deep, so I just want to give you guys like the pretty basic like principle like how we are thinking about like or design for like the drug deliveries. And if you are interested, that's a pretty nice like review papers on like the Nature BME. So first, I just want to talk about like the cell replacement therapies. And why we're talking about is because like for Taiwan diabetes, like there's more and more like the patients every year in the States and over in the world. It's a autoimmune disease. So that means like our like the immune cells will cannot recognize our own cells and attack it. And in the Taiwan diabetes patients, they're trying to start to attack the pancreatic cells. So in the pancreatic cells, that's a cell secreting insulin. And if the insulin producing cell was destroyed by our like immune systems, that means like the patient was starting to lacking the ability to secrete the insulin, and those patients will have the high blood glucose. So the current like the treatment is like the patient need to do like the insulin injections, not just daily, every meal. So every time when I having any food, they need to have like the insulin injections. And why I'm talking about like the glucagon injections, because like we know like insulin, what's the function for the insulin? It'll help us to lower like the blood glucose. But what about if we overdose the insulin? That means like we might have like the low blood glucose condition, and the hypoglycemia will cause or induce put the patient into pretty dangerous like the situations. So that's why like people starting to thinking about like after we have the pretty nice like the insulin like the for a drug, how about the glucagon? And another thing is like insulin injections. As I mentioned, like patient not just do the daily injections, that's every meal. So I just want to ask like, how many people likes to get shots? No, right? So that's why like those patients they're always trying to look for like any other new way to treat their like the disease. And every time when I talk about the research about like Taiwan diabetes treatment using the cell therapies, after the presentation, there's always the patient came to the stage and talk to me. They want to volunteer for the trial. Yeah. So if you can imagine, it's like that's why like there's a lot of like the Taiwan diabetes like nonprofit organization trying to put effort, trying to push on these like the methods to all these like the therapeutics. So as I mentioned in the beginning, what people are doing is like doing the micro encapsulations to encapsulate the islet cells into the hydrogel capsules. And the good thing for the hydrogel is the pore size will allow like the oxygens, nutrients, and also the insulin to diffuse in and out in the like allogeneic capsules. But also like the hydrogel capsules can protect the cells from the antibody or immune cells attack, because we know like that's a normal foreign body response. And people might talk as like, but currently we have the stem cell technologies. Can we just use the patient's cell to derive into iPS and differentiate into the islet cell again? But just don't forget, because like that's an autoimmune disease. So even you got like the same like the cells, the pancreatic cells from the patients, their immune systems will still recognize it as a foreigner and attack it. So that's why we need this method to protect the cells and let them to like help us to respond to the blood glucose. But as I mentioned, the problem is like easy to implant. It's hard to like retrieve the capsules. So for example, when I doing the research implant into the mice, after I need to collect those capsules, because I need to do the histology or any like the categorizations, I cannot even find any capsules in the like the mouse cavity very easily. I just need to use a lot of PBS or saline to wash out all the capsules. And usually it takes me like at least 15 minutes for one mouse to retrieve most of the capsules. So can you imagine if for one mouse, like we can just use a lot of like saline to just wash out the the mouse cavities, but we probably cannot use the same method for a human. So that's why like we are trying to develop like the macro device. So this is also like the research I did with my like colleague at Cornell universities. So we are thinking about like, because the sutures will be an easy way we can easy to get and easy to implant into the bodies. And in the beginnings, we didn't want to put the sutures in the device, because we're just thinking about like, we just make like the string of the like the hydrogel to encapsulate the cells inside the device. But the problem is like the hydrogel is, the mechanical property for the hydrogel is too weak. So it cannot survive during like all the like the implantations or surgeries. So that's why like we think about like, how about we just put those like the sutures inside the like the string like the hydrogels to support like the mechanical properties. So we can easily to let like the surgeon to handle this like the device with the cells and do a suture into the bodies. So that's the idea and how we do the like the cell encapsulations. It's like we twist like the sutures and embed it into like the calcium, like the solutions, and trying to like precipitate the calciums on the sutures. So we can just like using like the manufacturing, that's the concept for like the biomanufacturing. So we can use like automation, like the rotation, like the machines trying to like put into the string into like the hydrogel, like the solutions quickly. And at the same time, like when they roll into like the hydrogel solutions, the hydrogel and the cell will cross-linking and stay on the sutures. And this is the one video like showing like what's the difference when we use like suture as supporting like materials. You can see like on the bottom, that's just pure hydrogels. It's pretty weak. Even like the forceps can easily to break the hydrogel strings. So you can imagine like if the surgeon using the forceps to bring those like the hydrogel strings, it's impossible for them to do the suturing on the patients. And this is another video when we put those like the hydrogel strings in the mouse cavities, and when we collect them, it's hard to retrieve the hydrogels. And as you can see, like in these the videos, the hydrogel string is pretty big. And what's the reason? Because that's the minimum we can do for the implantations. Otherwise, we cannot handle it. So once we develop these methods, we just want to see how you can do or work in the large animals. So the good thing for working at Cornell, because we are pretty strong the vet school, so we have one vet help us to do the endoscopy for the dog surgeries. So that's the yeah. We can see we just let our the device on the livers, and after 1 month, we're trying to check if our material cause any fibrosis or any other immune response might damage our the device. So, but we're pretty lucky. So this material and the device work pretty well in the dog, and we can easily just use the endoscopy to retrieve our device to in the future, if we really want to use it in the human, we can easily to use endoscopy to replace the cells inside the Taiwan diabetes patient's body. Yeah. And okay, so that's inside the dog's cavities. So that's the outside. So how we do the surgery, that's pretty easy. We just have the same as normal endoscopies. We have several hole we can put into the microscope, and also our robot to put into our device into the bodies. And one more thing we are talking about is why we are checking the device after 1 month, because there's a potential problem for the fibrosis. So although the material is great, it can protect the cell from an immune attack, but our body also pretty smart. So if our body cannot attack those foreign bodies, another thing they are trying to do is trying to separate the foreign bodies from our bodies. So after 2 weeks, the immune system will release the cytokine and induce all the fibroblasts to come around in the implants. And then these fibroblasts will try to secrete a lot of the collagens. And these collagens will form the collagen fibers and cover the whole the implant. And that's how our body trying to protect ourselves. And this happens a lot in all different the implantations. So, and in our case, if the fibrosis cover the whole device, that means it will block all the nutrient, the mass transfer. So that means the cells will die. They cannot get any oxygen or nutrients, and also the insulin cannot diffuse out. So that'll be the new problem for us. And what we need to do is we need to find a better way, the better materials we can do for the anti-fibrotic, the properties. For example, here's the papers published from my previous colleague at MIT. So the Z2Y12, that's one material. That's a peptide we conjugate onto the alginate, and we found if we put this the peptide on the surface of the hydrogels, it can help us to suppress fibrosis formations. That means we can definitely use it for the cell replacement therapies in the future, or even for any kind of the medical implantations. There will be a way we can do trying to prevent the fibrosis. And as I mentioned, first testing doing a clinical trial. So they have 2 different trials. One is the fixed AAT. Let's just simply use the stem cell derived pancreatic beta cells to implant into the patients. And they but those patients need to use the immunosuppressant drug. And the result looks pretty good, but unfortunately, in early of this year, that 2 patient dies. And so this can try to on hold, but I think this still give us or give a lot of the Taiwan patients a huge hope in this the cell replacement therapies. And the second one is the 2VX, the 264, they are using the device to encapsulate the stem cell derived the beta-like cells and implant into the patients. So the good thing is the cells can be protected by the device, and the patient no longer need the immunosuppressing the drug. So this method can help us to help the patient to increase their the survival rates. Because allow patient die, the higher probability just because the bacteria infections. Okay, so the next topic I want to talk about in the drug delivery will be the mRNA the delivery. So for people not familiar with mRNA, so in molecular biology, we all know we have the DNA will transcribe into the RNA, and only the messenger RNA will translate into the proteins. So in the drug modalities, a lot of people talking about deliver the drug as a protein, a peptide. But what we are thinking at MIT is deliver a protein. So how about we just deliver the DNA or RNA into the bodies, let our body to produce those protein for us. So at MIT, in the beginning, we did both, but why we choose mRNA in the past 10 years? Because for the mRNA delivery, we just need to encapsulate let me use this one. We need to encapsulate mRNA into the lipid nanoparticles and find the right cells and trying to pass through the cell membrane, because we know cell membrane, the major component is the lipids. So we use a lipid as a material trying to go through the cell membranes. And once we enter through the cells, release the mRNA so we can use the ribosome to do the protein synthesis. That's not simple, but compared to DNA, we first need to overcome to go into the cells. The second thing is we need to go into a nucleus. So we'll have the 2 different the barrier for us for the DNA delivery. So that's why we choose the RNA delivery as our target, because we believe the mRNA will be good enough to help us for a different therapeutic purpose. So for mRNA therapeutics, there's 2 different components. The first is the mRNA. So that's also we call the drug or the drug payload. And in the mRNA, let's find the component pretty important for us. So mRNA have the coding regions. So we have to have the good coding optimizations, and so we can have the high the mRNA expressions. And the UTR, we have the 5' UTR and 3' UTR. Both play pretty important role, because in a lot of companies, even they have right coding regions, but they don't have the good a UTR that makes them still have a pretty low the mRNA expressions. And we also need a 5' cap to try to minimize the immune response, and the poly-A tails that our body knows that's a messenger RNA. And the second thing is there's a delivery vehicles. And for the mRNA, and especially for the COVID vaccines, we use a lipid nanoparticles as a vehicle to deliver mRNA. And because we believe the lipid, we use the lipid as a material will can help us to improve the penetration through the cell membranes. So that's why we use the lipid as a materials. And for the current lipid for the mRNA delivery, we have 4 different categories. One is the polymer-based materials, and people starting to interested in this because people are thinking about like degradable materials. So polymer-based system will be a major like the category for the degradable materials. And the ionized lipids here, we can see that's a different ionized lipid people use. For example, SM102, that's a Moderna's lipid, and ALC0315, that's a Acuitas lipid, but out licensing to the Pfizer's and BioNTech. And they also have the Intellias lipids. And the CKK12, it's a lipid developed by MIT. And first, out licensing for like the nylon for siRNA delivery. And after that, like we out licensed to like the Translate Bio, currently is acquired by like the Sanofi for the mRNA delivery. And also we have like the street RNA lipids and also the dendrimers. So all different lipid has their like the pros and cons. And while we are designed for the delivery, well, first we need to think about like what's our purpose, what's our indications, and what's our like the final like from a clinical aspect, like what's the administration. So all this will highly affect the how we design for the drug delivery for the mRNA. And in case not everyone here familiar with lipid nanoparticles, so they just the pretty like the introductions on like the lipid nanoparticles compositions. So we can use the lipid nanoparticles for nucleic acid delivery, for example, mRNA, siRNA, ASO, or even the DNA. And we need a help lipids. So help lipids can help us to provide like structural properties. And we have like different like help lipid. For example, in the COVID vaccines, both like the Moderna and Pfizer use DSPC, and other formulations, for example, our previous lab developed using the dope as a help lipid. And all different help lipid have with their different structures, we can see the different structure of the lipid nanoparticles by a cryo-EM. So when people ask me about like what's the actual structure for LNP, first thing I will ask is what's the formulation you are using? Because different formulations, you can see we can eventually form different types of like the structures. And we definitely can see the different structures by the cryo-EM here. And the major component for the LNP is the ionized lipids, because the principle to encapsulate the mRNA or DNA into the lipid nanoparticle is like we use in like the acidic conditions. So the ionized lipid will carry the positive charge and interact with the phosphate group of the nucleic acid to form the nanoparticles. Then we have the help lipid and also the cholesterol. Cholesterol is the major component, or also what we call is like structural lipids to help us to stabilize the LNP. And also another key component for us is the PEG lipids, because that's not traditional like the polymer-based nanoparticles. In the traditional polymer-based nanoparticles, people want the nanoparticle to carry like the positive charge or negative charge to prevent aggregations. So, but we don't want the lipid nanoparticle to carry like a lot of the charge on the surface, because we all know like the positive charge usually will induce a toxicity in the liver, and this will cause a lot of huge problem. So we put a PEG lipid to stabilize, prevent the aggregations, formations. So, but PEG lipid also have some problems, especially in Asia, because like there's a papers from the 中国医药大学. Then they are studying like the people in Taiwan, like if they have anti-PEG like antibodies, and they found like I think that's a 70% or 80% of the females in Taiwan, they all have the anti-PEG antibodies. So maybe that's one of the reasons like we see a lot of like side effects when people like receive the mRNA COVID vaccine in Taiwan. And another key component for the drug delivery for the LNP is like endosomal escape. And we have found like we developed a lot of materials at MIT, and some of them we believe that's a really nice like materials. We can see a lot of mRNA inside the cells because we tag the mRNA, but we didn't see a lot pretty high like protein expressions. And eventually we found out why, because we didn't release the mRNA inside the cells. So what people study is like the apparent like the PK for the LNPs, because what we want to do is like when the LNP enters the cells, because there's a pH change from like the 7 to around like the 6 or 5.5. So we want the lipid particle to respond to this pH change. So for example, our ionized lipid can carry a lot of like the RNA, like the or proteins inside the LNP, and this will form like the what we call like protein sponge and will increase the osmotic pressures. So a lot of water will go inside the LNP to break the lipid particle to release the mRNA inside. So there will be like another thing people starting to thinking about, like we are not just thinking about like the good encapsulation of mRNA, but also we need to find a way to release those mRNA inside the cells. So using one of my previous slides to just explain how we do like the drug delivery or lipid formulations like studies in the early stage of the research. So we can choose to hand mixing or using microfluidic device to form the lipid particles. And to me, because I skipped the in vitro like the assays because I didn't see any good like the in vitro models we can use that's translatable to in vivo. So I always just screen the LNP in the mice. So first thing I will do is like use the DOS to find out if this formulation can form or this material can form nice like single peak lipid particles. If it cannot, I won't inject into the mice. But if it did, I will just direct into the mice to do the LNP screenings. Then because for this project, I use the 5' luciferase mRNA. So after 24 hours, I use IVC sense to do the imaging, find out like, is there any like organ was transfecting, have the luciferase bioluminescent signals, or if this lipid is good for the mRNA delivery, and also like does it also induce any toxicities? Because for some like the case like after injections, and after 24 hours, when I digest the mice, I found like the mice have the internal bleedings after I digest the mice, dissect the mice. So that also gives us some ideas how we design the materials and what kind of formulation we can use for the LNP studies. Okay, so just talk about like the current trend. So people pretty familiar with like mRNA therapeutics will perfectly used for like infection disease. So COVID will be the great examples. And people, Moderna and Pfizer also developing the mRNA for the flu as well. And that's a paper I probably published last year. That's from like a Drew Weissman's lab at UPenn. They use the mRNA to do the universal like the flu. So they put any type of like the antigens and making into a different mRNA and just mix them and inject into the mice to make it like the universal like the flu shot. And second applications, people are the pharmaceutical company I think interesting about like, for example, here's like Moderna use the mRNA for like the tick virus like preventions, but this prevention not just like use the mRNA for the antigens, but instead they use the mRNA to produce the antibodies inside our like human bodies, so we can have like enough like neutralizing antibodies to protect us like by the virus. So this is clinical trials by the IV infusions, and to me it's still pretty good because like the dash line here, that's 1 microgram per ml. That's like the minimum like the antibody we need for prevention purpose. So that means like at least we can use this like the mRNA to protect us like at least for like a couple months. And another thing is like things like mRNA, just like the single pretty like quick like single peak like expressions. So people are thinking about like mRNA will be a good way for like gene editing. So that's Intellia therapeutics like the clinical trial, like they use the LNP and the AAV for the CRISPR like the gene editing purpose. And that's two different like the targeting they are doing in a clinical trial. And the last things, I'm also working on this product in my companies. But here is I just showing like the generation bias slide. So people are thinking about like the in vivo CAR T. So current CAR T is like we need to collect the cells from the patients and doing like the lentiviral infection for the CAR, like the ex vivo, and infuse back into the patients. That means like each dose is pretty expensive. And if we can just use the LNP to deliver the CAR mRNA, or even we use the DNA, but we just need to any targeting molecule to targeting the T cells in vivo. We can just simply make it cheaper and make it universal like a CAR T for everyone. So that's a generation bios data. In the mice, we can see like they show pretty high, like 60% of the T cell was transduced with the GFP mRNA. And even they increase the dosage, they can highly improve the whole like the gene delivery in the mice. Okay. So because I just want to think like because all my like the data or previous research at Cornell and MIT like at MIT I really think like Bob and Dan, and they are like the generous like support. Like usually I have like pretty unlimited like budgets in the research at MIT, and Tara was the one to, she's an administrative assistant, and she helped me a lot. It helped me to submit like a 2-hour grants and helped me a lot on all the administrations. And Dr. Han, she's the one helped me on the new materials like the synthesis, for example, the PLGA with the glucose for the glucagon delivery. That's she's like contributions. And thanks like Anna, Teresa, Luke, Ellen, and Dennis. And my collaboration at Just in the Epicenters, Yuhua, Zhihao, and Tatataga. And all my community members and colleagues in the lab at Cornell. And also thank all my like founding agents. And I'm happy to take any questions.